Hi everyone, and welcome to Unit 11, where we're going to do an introduction to parasitology. In case you're wondering, there are about 8.1 million people that have parasites, or about a quarter of the world population has parasites. There are not many parasites endemic to the United States. However, there are parasites endemic to other countries, and people travel so globally so easily these days that it's not uncommon to come across a parasite that's not native to this country. So stay tuned. Let's get on with this lecture in parasitology. Now, getting into parasitology, there are a few terms that you need to be familiar with. The first term is parasite. This is an organism that lives on or in another organism and requires something from the host. The host has something that the parasite requires for its existence. Then we've got parasitism, which is the state of being infected with parasites. Host is an organism which harbors a parasite. Usually the host is a human or an animal. We can break that down into further terms like definitive host and intermediate host. The definitive host harbors the parasite's sexual stage, while the intermediate host harbors the parasite's larval or asexual stage. Then we have reservoir, which is a temporary replacement for the host, and it may be the source of infection. And then we have commensalism, a symbiotic relationship or a close association between two different organisms. Both organisms benefit also. This may also be known as mutualism as well. Neither is harmed in commensalism, but in mutualism we have a symbiotic relationship where both benefit. And in commensalism we have a symbiotic relationship where one benefits, but one isn't harmed. Okay, a symbiotic nature is one of protection or nourishment. An endoparasite is one that lives within the body of the host, and I usually think of protozoans or the helminths. Then we have ectoparasites that live upon the body of the host, and again I think of arthropods like fleas, lice, and ticks. Then we have facultative parasites that can live as a free organism or as a parasite on the host. An incidental parasite, which is a parasite that lives with the host where it normally wouldn't live. An obligatory parasite, which is the parasite that cannot live apart from its host. And then finally we have vector, and usually this is an insect that transports pathogens from the infected to the non-infected host. For example, some vectors would be the mango fly or the Anopheles mosquito. And I promise this is the last slide on terms. Carrier is a host that harbors a parasite but is asymptomatic. An infection is invasion of the body by pathogens. An infestation is a little bit different. This is arthropods living on or within the host. And then we have protozoan cysts and trophs. A cyst is a non-motile, non-feeding infectious form of a protozoan. It usually has a hard cell wall. It is very rigid looking in appearance. And then we have protozoan trophozoites, which are the motile, feeding, and non-infectious form of a protozoan. Usually the trophozoite has flagella and it looks like it's motile. There's many types of specimens that are acceptable for parasite culture or parasite studies, Ova and Parasite studies. Most of the time what we see that comes into the laboratory is feces. But you can also see catharsis, which is a purgative cleansing of the bowels. Sigmoid or duodenal samples are also common. Then we are looking for gastrointestinal pathogens. In the urine, we could be looking for helminths such as Schistosoma hematobium or Trichomonas vaginalis. In the genital tract, we could be looking for Trichomonas vaginalis. Sputums, we're looking for the burrowing parasites. These are worms that go from the gastrointestinal tract to the respiratory tract, and that can be Ascaris, Strongyloides, Paragonimus. Those all have a lung phase. And then finally we have aspirates or biopsies. So they could be lung or liver or lymph node biopsies, spleen, bone marrow, muscle tissue, and cerebral spinal fluid. These are all aspirates or biopsies that we can look for parasites. But for the most part, usually what you see is a stool sample. So let's take a look at the requirements for feces, sigmoid colon sample, and duodenal sample. Okay. In the feces, if we were collecting that for ova and parasite study, we want to make sure that they've had no barium enema. We don't want any barium contamination. We do want the feces to be fresh, refrigerated, or preserved. It's best to preserve it, and refrigerating it is the second best option. And there's a number of preservatives we're going to talk about in upcoming slides. Feces, you want to avoid contamination with urine or toilet water because a urine pH will actually destroy fragile parasites. A urine pH is usually slightly acidic. Water from the toilet may destroy or alter osmotic fragility, or it may have its own parasites. Toilet water often has what's known as wireworms. Okay? And again, the most common type of sample that you're going to see is going to be fecal matter. Okay, so the sigmoid colon, this is a sample that is taken from the large intestine closest to the anus. It is examined when feces is negative for parasites. The specimen is obtained by aspiration or scraping. They scrape the tissue with a sharp instrument of some sort to get the tissue in order to view it. Cotton swab collections are a poor sample choice. We should never use cotton swab collections for sigmoid colon, ova, and parasite study. And then finally, a small volume is usually obtained for the sigmoid colon. Duodenal sections. The duodenal section is the first section of the small intestine adjacent to the stomach. Okay? We are going to collect those samples by using what's known as the string test. In the string test, we use a weighted gelatin capsule that is swallowed and then pulled out later at about 4 to 6 hours. Okay? Usually a small volume is obtained for the duodenal sample, and we can examine the specimen with a little bit of saline and with that sample being centrifuged. And we're looking for Giardia, which is a motile parasite that we look for commonly in the duodenal samples. A catharsis sample is a forcitive washing of the cecum. That's the first part of the large intestine near the appendix. With this purgative action on the bowels, we can induce it via saline, and we're looking for cysts. Cysts are more likely than trophs to be seen because cysts can tolerate the forcitive washing of the cecum, where trophs are very fragile. If we're going to examine urine and genital samples for ova and parasites, we want to make sure that the urine sample is a clean catch sample, and that we want to make sure that the sample is fresh and that is centrifuged and that we look at the sediment. Usually in urine samples, we're looking for a trematode bladder fluke known as Schistosoma hematobium, and this is associated with wading in water in Asia and Africa. For genital samples, we're often looking for Trichomonas vaginalis. We can collect prostate, urethral, or vaginal secretions. And we can collect swabs and add sterile saline to the swab and examine by using the wet prep method. Again, we're looking for Trichomonas vaginalis, not to be confused with other Trichomonas species that are GI pathogens. Sputum samples, aspirates, and biopsies can all be examined for ova and parasite studies. Okay. Sputum samples, we want to collect the first morning sample upon waking. Usually that is our most purulent sample, and we do want to collect a deep specimen. We want something that's coughed up and not just spit or saliva. Okay. Some parasites actually have a lung phase where they pierce through the tissues and invade the lungs. We want to view wet preps or permanent stained slides, and we're looking for things like Ascaris. In aspirates and biopsies, these surgical biopsies are best for lung, liver, and lymph node samples. Here we're looking for lung flukes or liver flukes. Cerebral spinal fluid and body fluids can be examined, but we do want to make sure that the sample is concentrated, especially if we're looking for neurological amoebosis, which is basically a fancy way of saying Naegleria fowleri is an amoeba that can cause neurological problems. Okay. We do want to perform wet preps on aspirates or biopsies, or you look at the stained permanent slide as well for ova and parasites. Now, if a specimen comes in for ova and parasite study, it's important that if we cannot look at it immediately, that we place it on a preservative. And there's a number of different preservatives that I'm going to talk about today. 10% formalin, methylate iodine formaldehyde, polyvinyl alcohol, sodium acetate, acetic acid formalin, Schwaden solution, and Parasafe solution. So refrigeration is a second best option, but putting the stool on a preservative within the first hour is the best option for preserving that specimen. Here we can see many vials or examples of preservatives. Some are good for concentration exam, and some are good for permanent slides, and some are okay for both. But part of the O and P examination would include the gross exam, that's the macroscopic exam, the direct exam, that's looking for motility and any kind of morphology that we can see on a raw stool. Concentration exam was where we concentrate the specimen, and finally the stain or the permanent stain, which is usually a trichrome. Not always is every part done. Okay, so if a specimen comes in and it's not on a preservative yet, or it's already on a preservative, we cannot do the gross exam or the direct exam. We can only do the concentration and the permanent slide stain exam. Okay, so let's move on to the next slide. So some stool preservatives are going to be better than others. 10% formalin is a really nice preservative for looking at our concentrated samples. Okay, so it's a good preservative for concentration procedures. The disadvantages is that it's not suitable for permanent stains, and it's inadequate for preservation of trophs. So in other words, we can save cysts on 10% formalin for years, but the trophs will break up. Then we have myriothiolate iodine formalin. The advantage is that it's a good preservative for concentrations. It's easily made and prepared in the laboratory, so that's not a problem. Some disadvantages: it's not suitable for permanent stains and is inadequate for the preservation of trophs, and the iodine is unstable and may cause the protozoans to look kind of funny. So that's MIF. Polyvinyl alcohol, or PVA, is another one that I see commonly in the laboratory. Some advantages: this preservative is good for the preservation of cysts and trophs. It allows the specimen to be shipped worldwide, and it preserves these samples for months or even years. Okay. A disadvantage is that it's not good for direct exam or concentration, but that's okay. We can use 10% formalin for that. And it does contain mercuric chloride, which is a carcinogenic. Okay. Sodium acetate formalin, or SAF, is another preservative. The advantages are that it's good for concentrations and permanent stains, and it's even suitable for acid-fast stains. The disadvantages is samples will not adhere to the slides very well, so we have to add a little albumin and glycerin to the sample so it doesn't slurp off when we go to do our staining technique. It's really not a good preservation preservative for trophs either. Then a few more preservatives. We have a Shodden solution and Parasafe. Shodden solution is a good preservative for cysts, trophs, ova, and larvae. It's okay for direct exam and permanent stains, and it's okay for concentrations too, but others are better. Some disadvantages of Shodden solution is it's not good for concentrations, and again, if you have mucoid samples, they have a hard time adhering to the slide. The next preservative is Parasafe. The advantage is that it's good for direct exam and concentrations and perms. It's non-toxic and non-carcinogenic, but the advantage is it is very expensive. So basically what I've talked about is PVA, SAF, Shodden solution, Parasafe, and a few other preservatives that are good for the preservation of ova and parasites. Whenever we take in a stool sample for ova and parasite study, we always want to first start out with a macroscopic and a microscopic exam. So the macroscopic exam is also known as the gross exam, and here we are looking for worms and mucosa and proglottids. Proglottid is a segment of a tapeworm in the cestodes that harbors the reproductive male and female parts. That's a proglottid. We want to note if the stool is dark black or bright red. That could indicate blood in the stool. We want to note the consistency of the sample. Is it formed, semi-formed, or liquid? Liquid stools must be examined in 30 minutes. Soft stools must be examined in 1 hour. And solid stools usually need to be examined within 24 hours. We always want to note the date and time of the collection as well. For the microscopic exam of feces, we want to do a direct wet prep from the actual stool sample itself. We want to use saline and iodine. The reason we want to use saline is that's going to help any morphology and motility, help us observe any morphology and motility of any parasites, whereas iodine is going to make the internal structures of our protozoans especially pop. After we do a direct wet prep, and again, that may or may not be included, if we don't get the sample, if we don't get a raw sample, that's not going to be done. So sometimes the samples that we get in are on preservatives. And if they're on preservatives, we skip the direct sample, the direct wet prep, and we go right to the concentration. So the concentration, in the concentration, the sample is washed and then viewed on 40X, preserved, spun down, and the sediment is examined. Okay? Now we're not going to use gram iodine to look at this. We're going to use what's known as D'Antony's or Lugol's iodine. And then finally, the fourth stage of a ova and parasite study is the permanent stain, where we can see enhanced morphology because we're viewing slides on 100X. And there's two types of permanent stains. One is called the trichrome, and one is called the iron hematoxylin stain. Those are going to be viewed on oil, and we can either do that in our own lab or we can send them to a reference lab. And what's great about these slides is they last forever, and we can use them to study or use them for teaching, which is really nice. For an ova and parasite study, we always want to do the direct exam if we have access to the raw stool. If we have access to raw stool within the first hour of collection, we never want to add tap water or stellar water or anything like that to the sample because our trophs are very fragile and they might be destroyed. Sometimes found in tap water, we can find things that look like parasites but actually aren't parasites, and those are called pseudoparasites. And we'll get a chance in lab to look at all kinds of artifacts and pseudoparasites. Keep in mind, any tap water that's added to a raw stool could have its own parasites in the sample as well. And we do want to perform a direct exam on a fresh, unpreserved specimen, and we're looking for motility and morphology. We're looking to see if we see any motile trophs, and we're looking to see the morphology of any protozoans or helminths. The concentration is the third part of an ova and parasite study, and it's a method that we can use to separate parasites from fecal debris. And there's a number of different methods that we can use. One is called floatation, where the parasites float to the top, and one is called sedimentation, where they sink to the bottom. Okay, concentrations allows for us to detect even small numbers of parasites because we're spinning them down in the centrifuge. Cysts hold up well during this process, but trophs can be compromised because they are so fragile. There's two types of concentration methods. One is called the flotation method and one is called sedimentation method. It's usually the sedimentation method that is used in clinical microbiology, but in flotation we're looking for cysts and ova to be separated using a liquid with a high specific gravity, and they will float to the top. This is not a good procedure for heavy helminth ova because they're going to sink to the bottom. Usually our liquid with a high specific gravity is zinc sulfate with a specific gravity of 1.18 to 1.20. Okay, sedimentation I think is a little bit better. It's good for most ova and protozoans. Now the debris is not going to be eliminated as well as it would be in the flotation method, but it's still not bad. The cysts look less distorted than with flotation, and the trophs could be compromised because they are so fragile, and in a spinning centrifuge they may not hold up to that. Concentrations of sedimentation procedure can be kept for a long period of time. And there's many years that I sometimes keep these for. So there's two types of concentration methods. One is called flotation and the other is called sedimentation. So flotation requires the use of a liquid with a high specific gravity. Usually that liquid is zinc sulfate, and it has a specific gravity of 1.18 to 1.20. The ova and the cysts usually have a specific gravity of about 1.05 to 1.15. They are lighter than the specific gravity of the zinc sulfate, so they're going to float. Those ova and parasites are going to float to the top. Objects over 1.15 in a specific gravity value are not concentrated with this method. Zinc sulfate is used more common in the flotation procedures. So here's the flotation procedure. We're going to take a stool sample and we're going to put it in a test tube, and we don't need very much. We just need about a pea-sized portion, which is about a half a teaspoon of feces. And then we're going to add 1 to 2 mils of sterile water. We're going to centrifuge at 1500 RPMs for a minute and then discard that supernatant just into the sink with copious amounts of water. Then we're going to add a couple of mils of zinc sulfate to the sediment and resuspend that sediment by giving it a little shake. Fill the tube within 2 to 3 millimeters of the rim with zinc sulfate. Strain the suspension through gauze into a paper cup, then discard the gauze. And the reason we do that is we're looking for proglottids and scolex and other parts of worms. Okay. Return the suspension to the tube and add zinc sulfate again to within 2 to 3 millimeters of the rim. Then we're going to centrifuge it again at about 1500 RPMs for a minute. Then what we're going to do is remove a little bit of that surface film, just a couple drops, and we can look at those drops for ova and parasites. Add a cover slip and microscopically examine for those ova and parasites. So that's the flotation procedure. And again, this flotation procedure is not really a good procedure for very heavy helminth eggs. The sedimentation procedure for concentration is probably more often used than flotation. Ova and cysts will sink to the bottom and can be found in the sediment. Ova and cysts are separated from debris by sedimentation with centrifugation. So basically we wash the stool sample with a little bit of saline and some formalin and ethyl acetate, and what that does is it concentrates the sample, and so the stool and the ova and parasites will be at the bottom of the tube. Okay, so this technique, like I said, uses formalin and ethyl acetate. You got to have good airflow in the laboratory because ethyl acetate is basically like ether, so it can kind of give you a headache and make you a little sleepy. The sedimentation concentration method is good for all ova and cysts. The cysts are more normal in appearance than that of flotation. And formalin actually fixes the organisms and the internal structures. So you can actually see the internal structures of the cysts and the trophs a little bit better because we've fixed them with formalin. Now, sedimentation does not have to be examined immediately, where flotation does. In flotation, the ova and parasites are going to sink over a period of time. In sedimentation, the ova and parasites are already at the bottom of the tube, and we can save these samples for years to be looked at. So this sedimentation procedure is what we're going to be doing in our student laboratory. Basically, we have our samples that are already in formalin, and we're going to filter that through a piece of gauze. And again, we're filtering so we can find any proglottids or scolex or any parts of worms in that stool sample. We're going to take about 1 to 2 mils of sample that has gone through the gauze and put it into a 15 mil centrifuge conical tube. That's a tube that's kind of angled at the bottom. We're going to add about 12 mils of saline with a squirt bottle to that tube. We're going to cap it and centrifuge it at 1500 RPMs for about 2 minutes. After the 2 minutes has elapsed, we're going to decant the supernatant and then add more saline and resuspend that fecal button. We're going to cap and centrifuge again for 2 minutes. Decant the sample, and then we're going to add 7 to 10 mils of 10% formalin to the conical tube, and then we're going to also add 3 mils of ethyl acetate to the tube. Basically what happens is after you add the ethyl acetate and the formalin, you end up with 4 layers after you spin it down. You end up with an ether layer at the top, or ethyl acetate. Then you'll end up with a plug of fecal debris. And then below that, you'll have about 7 to 10 mils of formalin. And then finally, at the bottom of the conical tube, this is where the sediment is at, and that's where you're going to find the parasites. Okay? You have to rim that debris around the conical tube to get it to pour out when you decant. Then after you've decanted, after the last centrifugation, you're going to add just a few mils of formalin to that sediment, and you're going to be able to examine it with a drop of iodine or a drop of saline. So that's the sedimentation procedure. It is the most common procedure used to concentrate stool samples for ova and parasite study. And then here on this slide, what I've tried to do is just kind of outline the advantages and disadvantages of flotation versus sedimentation. So I'll let you take a look at that on your own. Some advantages of flotation is that it's easy and it requires no ether, which is a nice thing. The disadvantages is that you always have to be checking your zinc sulfate to make sure the specific gravity is where it needs to be. It's not good for heavy ova because heavy ova sink to the bottom, and you have to examine it ASAP or all those ova and parasites start to sink to the bottom as well. Cysts may look a little bit abnormal and wackadoodle. Now, in the sedimentation procedure, some advantages is that it's good for ova and cyst examination. The cysts look normal, and the sediment is stable for years. A disadvantage of the sedimentation is that it requires ether and formalin, both of which can kind of give you a headache. It's not necessarily as good for iotamoeba or Hymenopus nana. However, I've not found that to be the case. And you do have more debris to deal with in sedimentation. We can't quite get all of that sedimentation, all that debris from the bottom of the tube. Now, sometimes our O&P preps don't look so well, and there could be a number of reasons why that can happen. The smear can be too thick or too thin, and you should be able to read newspaper print through it. So when you add your stool sample to the slide, if you add iodine or saline, you want to be able to have it not so thick that you cannot read newspaper through it. The iodine has to be the color of dark iced tea. If it's too weak, too strong, or too old, this is going to cause you to not be able to see the internal structures. And actually, if the sample or if the iodine is too strong, you may see crystal clumps, or this may obscure parasites. The centrifuge speeds for concentration and spin times during concentrations must be monitored. We do want to make sure that our centrifuge speeds and spin times are appropriate. We do have to periodically check the specific gravity of zinc sulfate in the flotation procedure. Also, we want to make sure that we didn't use too much fecal material when we added it to the concentration initially. Again, we only want about 1 to 2 mils of fecal material at the most. You want to make sure that when you're doing a permanent stain, like with the trichrome, that all your staining times are correctly followed. And you want to make sure that the reagents and stains were made correctly. There's one part to the trichrome stain where we use acidified acid or acidified alcohol, and that we have to make with glacial acetic acid. We also make our iodine and reagent alcohol mix as well, and that has to be the color of dark iced tea. You have to ask yourself also, were the reagents or stains contaminated with tap water? Because if they were contaminated with tap water, this can actually cause a problem when viewing your permanent slides. Okay, now the trichrome, we're going to talk about that later in the lecture, but the trichrome is basically a 9-step process with different times required at each step, and it is a permanent stain where we can see lots of different contrast on the slide when it comes to ova and parasites and other debris. Let's go ahead now and look at some pseudoparasites versus parasites. A pseudoparasite is basically an artifact. And if you look here on the top image, you can see a budding yeast, and about the same size of that yeast on the bottom is Cryptosporidium, which is a parasite. And I will tell you, Cryptosporidium is very difficult to identify on a wet prep. The best way to identify Cryptosporidium is by doing a modified acid-fast stain, and it's because they're about the size of a yeast or a white cell, so they can be very difficult to identify in a wet prep. Here I'm comparing Entamoeba histolytica to white blood cells. You can see they're about the same size. Entamoeba histolytica is about 12 or 14 microns in diameter, and that's about the size of a white cell. Now, with a white cell or a macrophage, you can see the nucleus on the inside of the cell like you can on the right image. On the left-hand image with Entamoeba histolytica, you can see a cigar-like structure on the inside of that organism, and that's actually a chromatoid body. Okay, so you can see Entamoeba histolytica and white blood cells are very similar to one another in appearance. Another artifact that you can confuse with a parasite would be a squamous epithelial cell. A squamous epithelial cell is a little bit larger, maybe about 18 to 20 microns, and it has a small nucleus about the size of a red cell. If you compare the squamous epithelial cell on the left to the Entamoeba histolytica troph on the right, you can see that they look very similar. The thing about the Entamoeba histolytica on the right is you can see that it has peripheral chromatin around the edge of the nuclear membrane. So that's one way you can tell the difference between a squamous epithelial cell and a parasitic troph. Now, identifying worms can be a little bit difficult. One parasitic larva that you will see in a stool sample is called Strongyloides. It looks very similar to some of the hairs that come off the fruits that we eat. You can see on the left we have a peach fuzz hair. And then on the right, we've got Strongyloides. The difference being Strongyloides shows internal structures like a buccal cavity, which is the mouth, and you can usually see the esophagus of the parasitic larva as well. Plus, parasitic larvae, especially Strongyloides, are quite a bit larger than peach fuzz hair. A Strongyloides is about 200 to 400 microns, where a peach fuzz hair is much smaller than that, usually about 100 microns or so. Now, pollen can be problematic as well because pollen can appear many different ways. There's all kinds of pollen. There is one particular pollen, especially the one that you can see on the left here, that looks very similar to a tapeworm ova. The Taenia ova on the right is about the same size as the pollen granules on the left, and you can see that the Taenia ova has a thick cell wall around the worm, and you can actually see that it has radial striations. Pollen can do that as well. A lot of times pollen is trilobed, and you can kind of see that on the left-hand image. If you look at the very most left-handed pollen in the image there, you can kind of see that it's trilobed. Other things that we don't want to confuse with parasites would be things like oil droplets, starch or talc from your gloves, air bubbles, cheese mite eggs, which I will show you in lab, but they're very large, and most people have cheese mite eggs if they eat cheese, veggie spirals, plant hairs, plant cells, fungal elements. Those are all things that you may see on a fecal wet prep, but you want to make sure that you don't call those parasites. And then another thing that you may see are miscellaneous bugs, critters, and anything actually ingested by the patient. You can see an adult louse over there on the right, and I've actually seen lice in other insects in patient stool just because they ingested them. One other thing you may see is Blastocystis hominis. This is a protozoan, but it's usually not considered a pathogen. It's usually considered normal flora unless we have Blastocystis hominis in moderate to large numbers, and then it would be considered a pathogen. So let's take a look at the next slide about Blastocystis hominis. So Blastocystis hominis is not something we typically report unless we see moderate to many present. They are common flora among many people, and the pathogenicity of this organism is questionable. Their size ranges from 6 to 40 microns in diameter, and there is a central vacuole that pushes the 4 nuclei to the periphery of the cell. You can see in the top image on the right an example of a blastocystis that has been stained with iodine. You can see in the middle there that we have a large central vacuole, and then the dark dots on the exterior are the nuclei that have been pushed to the edge. If you look at the bottom image, this is an example of blastocystis that has been viewed with saline, and again you can see a large glycogen vacuole in the middle, and you can see how the nuclei have been pushed towards the edge. This is Blastocystis hominis. Charcot-Leyden crystals come from the degradation of eosinophils. Eosinophils, of course, are associated with parasites. If you see Charcot-Leyden crystals, you do want to report them in stool samples or sputum samples. You can actually see Charcot-Leyden crystals in people's samples that have parasites. They're highly associated with parasitic disease and hypersensitivity reactions. This indicates an immune response is taking place, and you want to look carefully at the samples for parasites if you see Charcot-Leyden crystals. So the top image on the right is a saline wet prep of a Charcot-Leyden crystal. You can see that it's kind of an elongated diamond shaped, and then on the bottom is an example of a trichrome stain for a stool, and you can see the Charcot-Leyden crystals are staining kind of purple, and again it's the shape that gives it away. They are large, kind of diamond shaped, skinny looking crystals. There are many signs and symptoms that a person has a parasitic disease, okay? If they have cramping, abdominal pain, they may have a parasitic disease. If they are anemic due to the hemolytic process from organisms like Plasmodium, that would be the organism that causes malaria, that could indicate that you have the malaria parasite. If you have appendicitis, it could be that you have a giant intestinal roundworm balled up in the appendix. That's called ascaris. If you have asthma or bronchial maladies, it is possible that some of these worms with the lung phase has gotten into your respiratory tract. If you have dermatitis, that is a common symptom of somebody that has parasitic disease. Diarrhea is probably the number one symptom of having a parasitic disease. That's especially true with protozoans. Okay, you could have edema and elephantiasis. This is true if you have a microfilaria infection in the lymph nodes. You can have eosinophilia, where 15 to 50% of your cells in your peripheral smear are eosinophils. That usually indicates that you have a parasitic disease. You can have fever and chills. That's very common in malaria, that fever and chill cycle. You can have no appetite and weight loss, and that usually has to do with tapeworms. And then finally, you may have suprapubic pain, which would indicate you may have bladder flukes. So all of these signs and symptoms are those associated with parasitic disease. So when you're treating your patient, we have to determine if they are dealing with a bacterial infection or a parasitic infection. And there is a number of things that we can do to kind of determine that. Okay. In an amoebic infection, there is scant cellular exudate, so you're not seeing an abundance of cells. There are more mononuclear cells instead of polymorphonuclear cells. You're going to see Charcot-Leyden crystals. Your RBCs can be increased, especially if you have something like a hookworm. And then protozoans like E. histolytica could be seen on the examination. If you have a bacterial disease such as Salmonella or Shigella, you could have an abundance of cellular exudate. You're going to see a lot of white cells, especially polymorphonuclear white cells. You're going to see, you're not going to see Charcot-Leyden crystals. Red cells may or may not be present, and protozoans will be absent. So that's a good way to determine if we have an amoebic disease or a bacillary disease. Now, when looking for ova and parasites, we always want to locate suspicious objects on 10x, but I will tell you the protozoans are pretty small, so it's almost better to identify them and find them on 40x. Your bigger helminth eggs can be found on 10x and then identified on 40x. We do want to adjust our condenser so objects are refractal. In other words, you may need to lower your condenser to get the best contrast. You always want to scan the entire slide and give each slide about 10 to 15 minutes of your attention. Look for things that are modal, especially if you're looking at something from a raw stool. And then we always want to use low light. Anytime we are looking at a saline or an iodine prep, we want to use low light. And that means closing your iris diaphragm and maybe even lowering your condenser. So that's it for Unit 11, Introduction to Parasitology. If you have any questions, please reach out.